Yeah, you. A little closer. Come on.
So you can talk.
Yep.
I feel like I would have watered you more if I knew that.
No, you wouldn't have.
Yeah, probably not. So, plant.
Okay, rude. My name's Vanya. Thank you very much.
Sorry, Vanya. What do you want?
Well, I want you to help Terry.
Okay, who's Terry?
If you must know, Terry's a turfgrass system from New England. He's been having a real tough time with some dollar spots, and he asked if I could help.
What are dollar spots?
So, dollar spots are these yellow areas in a lawn, sometimes caused by this genus of fungi called Clarireediae. Terry has been having a real tough time with this particular species, C. homoeocarpa, and he just really needs some help.
Okay, so then just use fungicide. Don't start randomly talking.
All right, genius. We tried fungicide, okay? It doesn't work. It's resistant.
Okay, that sounds rough, I guess, but again, what do you want from me?
Well, there was this lab at the University of Massachusetts, right? They collected samples of his fungus, tried to figure out why it was resistant to the fungicide. And Terry was looking through the data, and he thinks he sees a viral sequence in the genome, but he's not sure if it's a virus that's infecting the fungus and whether he could do something with it. And he wants me to take a look.
Okay, then just do that.
Well, if you haven't noticed, I can't exactly type, now can I?
Okay, fine. I'm going to my desk.
Hey, take me with you. Okay, Terry gave me this palm print sequence that he found in the genome, and then he said to use SRR 5980381 from the Bio Project from the University of Massachusetts, since it had the highest coverage for that particular palm print, at least according to her Serratus.
Okay, first things first, let's submit the palm print to BLAST-P and see what we get.
Wait, all of the BLAST-P results are for mitoviruses. It's probably mitovirus then.
Yeah, I think you're onto something, but we need the whole sequence. Let me try using tBLAST-10 with the palm print and then the SRA number that Terry gave us to see if I can get the whole thing and run it through ORFinder. Okay, so I found the contact with the palm print sequence, but when I run it through ORFinder, all of the resulting sequences that it gives me are like 75 amino acids, which is really small for like any significant proteins. And I don't think it's what we're looking for. I don't really know why.
Yeah, that's too small for an RdRp. Since it is a mitovirus, and mitoviruses are a species of viruses that infect mitochondria, wouldn't it use the genetic code of its host mitochondria? And if we're guessing that its host is this fungus, shouldn't it use the fungal mitochondria code?
Yeah, you make a point. Let me try that. Okay, so when I try genetic code 4, which is for the mitochondrial code for fungi, I get an open reading frame that's 793 amino acids, which that's really unlikely for it to be a coincidence.
Yup.
Okay, so when we go through BLAST-P, it shows that it's an RdRp, and since mitoviruses are really small genomes, we probably have the whole genome. So now that we have that, I think I'm going to try submitting the sequence with other known mitovirus RdRps and to create a phylogenetic tree using ng phylogeny, which is just this like workflow. And then I'm going to submit it to AlphaFold and then FoldSeek to see if anything interesting comes up. I'm going to go do that. I think I got it.
All right, let's see it then.
Okay, so when the results of the phylogeny tree showed that like our novel virus, which I've called like Fungicola clarireediae, which just means like little fungus dweller that lives in Clarireedia. Well, all of the results are showing that it's clustering with species that infect pathogenic fungi, which I think means that it's probably infecting that fungus that's bothering Terry.
Yeah, you're gonna need more to convince me of that.
Okay, yeah, you're not convinced, but that's not all I have. Check this out. So you know how I submitted that sequence to AlphaFold? Well, I took the structure that AlphaFold gave me and then submitted it to FoldSeek, right? And the top 2 alignments that I got were both for mitoviruses that infect pathogenic fungi. And their TM scores are around like 0.86 and their distance is pretty small. So like, I'm pretty confident that the structure is pretty similar, which I guess is more evidence showing that it's infecting that fungus.
Okay, but look at the top right corner. That's not very similar.
You mean the top right corner where AlphaFold wasn't even sure?
Wait, so then if you found this mitovirus that infects this fungus, couldn't Terry just use it instead of the fungicide? This is great. Okay, leave.
I'm putting you outside.